G'day nerds, ready for a nice relaxed start to the video? We're going to start with scientific variables today. Um, Basically, these are the components which make up our experiment as scientists. These are kind of our main ideal, right? Like this is our main core area of concern. So by the end of this, you should be able to identify the types of variables that exist within an experiment and what their purpose is, what it is that they do. So here's your vocabulary. If I can just get you to pause the video for a minute, write this down, and you'll have to refer back to as we move through the lesson. So first, let's look at an experiment. What determines the period? Now that's the amount of time for a complete cycle, a complete swing backwards and forwards of a pendulum swing. This here is a pendulum. That's straightforward, right? That's our pendulum. And what is it that determines the time? There's two options that I can think of off the top of my head. There's heaps of options I can think of off the top of my head, but there's two we'll start with. We'll start with the mass of the pendulum, so how heavy it is. So we measure that in grams. Or the length of the pendulum, which we would measure in meters. We could measure it in centimeters, probably should measure it in centimeters, but meters is the unit that we use. So first off, with our experiment, let's look at control variables. Control variables are constant and not varying. In other words, they would be variables if we didn't control them in the first place. So a valid experiment has one independent variable, one dependent variable. I know I did two fingers there, I meant one. One independent variable, one dependent variable, and everything else is a control variable. They are kept constant or the same, and this is so that they don't affect the result. So we know what is actually causing the change. They allow us to make an accurate and fair comparison. We call this a fair test, right? So this makes sure that, again, the only thing affecting the change, which is the dependent variable, is the independent variable. So our experiment needs to become two experiments because we've got two possible things. That's two independent variables, so we split them up into two things. Because if we changed both the length and the mass in the same time to see which has an effect, we wouldn't be able to work out which one it is. But that's why we're going to stick with this. So we break it into two experiments, and the independent variable for one becomes the control variable. So the one that we change becomes a control variable, kept the same for the other experiment. So to find if the mass affects the period, all other variables, such as the length, become constant. To find if the length becomes, so the length becomes a control variable, sorry. So if you want to find if the length is what's changing it, then the mass needs to become a control variable. Everything else are also control variables. The angle of the drop, the type of string, type of knot, what we tie it to, so forth and so forth. Now we talk about the independent variable. And we've just discussed we now have two separate experiments to work out our two separate ideas. And the independent variables are the ones that I change. So the I, independent, also known as the manipulated variable. So changed by the experiment. They are the one that we are changing, and we're going to measure the effects later on, but they're the one we're changing. So we're testing it to see what its effect is. So the first experiment, our independent variable, that's what IV stands for, our independent variable is the mass of the pendulum, okay? And our second one, our independent variable, is the length of the pendulum. Two separate experiments with two separate independent variables. They need to be conducted separately. Everything else is a control variable, but our independent variable is the one that you change. Now, this is important. When we collect our data, we'll put it in a table, most likely. The independent variable goes on the left-hand column. And when we graph that data, because we're scientists and we like to make graphs, we graph it along the x-axis of the graph. So independent variables go in the left column of the table and on the x-axis of the graph. So again, we've got our two experiments, right? The dependent variables are the ones that change depending on the one that I changed, okay? That's also called the responding variable. That's actually the way I like to think of it. It responds to the change in the independent variable. And this is the variable that we measure. So you don't measure the independent variable, you set that up, right? You'll have to measure it out, but you set it up. You measure the response in the dependent variable. So, there are two experiments, right? In experiment A, the dependent variable is the period, But in experiment B, the dependent variable is also the period. So the dependent variable goes on the right-hand side of the column. So independent, then dependent. When we put them in tables. And when we graph it, as I said, that's what we like to do, we graph our independent variable along the x-axis, and our y-axis is for the dependent variable. So when we talk about experiment A with the mass, mass A might be 10, the period is like 0.4 seconds, mass 20 goes along this way, and the period will be about 0.4 seconds. Mass 30 grams will be here, and then so forth, still about the same spot. So we go across for the x-axis with the independent variable, and up or down with the y-axis for our independent variable. All right. I hope that made a lot of sense. If you have any questions, feel free to put them in the comments below. We'll get back to you as quick as we can. Thank you for watching, and yeah, we'll catch up with you next time. Bye now.